Hi, my name's Tina Nguyen, and this summer I conducted research in the Beta Lab. And for my presentation today, I'll be talking about the systemic pesticide transfer from treated corn to wild mustard poses a risk to non-target herbivores. So modern agriculture relies on chemical treatments to protect crops from insects, fungus, and pathogens. Unfortunately, these chemicals can linger in soil and water, which can leach off to neighboring ecosystems and affect non-target species. Neonicotinoids, a class of insecticides, actually has been linked to pollinator decline. So in response, New York has passed a bill preventing the use of prophylactic usage in neonicotinoids on major staple crops. As alternatives, diamides are being considered as they're generally less toxic to pollinators, but we still don't fully understand its potential indirect effects to the environment or non-targeted species. So my research focused on two questions. One, do fungicides and insecticides on treated plants affect adjacent plants' growth and secondary metabolites? Two, do these treatments also affect non-target herbivore performance? So in my experiment, I started off with growing wild mustard adjacent to treated corn. And from there, we did three things with the wild mustard. One, we measured its height and leaves weekly. Two, performed an insect bioassay with the fall armyworm. This is to measure the relative growth rate to assess herbivore performance. And third, I wanted to quantify plant defenses by looking at secondary metabolites. We did this through liquid chromatography mass spectrometry. And so for our results over here, in our first figure, this is looking at the wild mustard height and leaves on all treatments. And we see that it's not been altered by the treatments throughout all 4 weeks. For figure 2, this is looking at the wild mustard flower and leaf secondary metabolite profiles. And we also didn't see any significance across all 4 treatments as well. Figure 3 is our relative growth rate in our fall armyworm feeding on the wild mustard leaves. Figure 3A is actually preliminary data from my mentor. We see that the diamide treatment right here actually decreases the relative growth rate of the fall armyworm. But in my data, in figure 3B, I didn't see any of changes across the board. Figure 4 is also relative growth rate, but this is feeding on flowers instead. Preliminary data shows that there is treated fungicide increase in relative growth rate. And for my data, also did not see any significant difference. So interestingly, previous studies have been shown that pesticides actually do alter plant growth and plant defenses. But in my case, I didn't see any of those patterns with the wild mustard and the caterpillar growth. So in future directions, we like to see if we repeat these experiments with a controlled seed source, or we like to continue evaluating if systemic pesticide exposure actually does affect non-target herbivores. Thank you.
Were you looking at this in the field or in greenhouse conditions?
This is in the greenhouse. So when I planted these, they actually were in a pot, like all across.
Thank you.